Hello, everyone, and welcome to today's seminar series called Cutting Edge Advances Towards Personalized Mental Health, hosted by our Center for Precision Mental Health and Wellness. I'm Dr. Adam Pines here with co-director of the seminar series, Dr. Chue Zhang, and I'll kick off with just a couple of housekeeping items. First, this webinar is being recorded and will be posted on our website next week. You can find our website in the chat box or scan the QR code on the left side of the screen. From there, you can sign up to receive announcements about our future seminars and events. If you're interested to join our center as a research member, please see the link in the chat or scan the QR code on the right-hand side of the screen to fill in a survey form, and we'll get back to you. And finally, please put your questions in the Q&A box, and we'll attend to those throughout, and then get to them at the end of the presentation. I encourage you to put your questions as early as possible so we can address them in that order. And for panelists, if you have a question, please raise your hand. Now, I'd like to introduce the director of our center, Dr. Leanne Williams, to introduce our special keynote speaker for the day, Dr. Desmond.
Let me unmute myself. Thank you to you and Chue for co-directing this symposium. Welcome, everybody. It's an absolute delight to introduce Dr. Desmond Oates, a highly respected colleague in our field. Dr. Oates is an associate professor of psychiatry at UPenn, and he directs the UPenn Center for Brain Imaging and Stimulation. Some of you may recognize Dr. Oates from his captivating plenary at the Society of Biological Psychiatry last year. Had the pleasure of being there, as several of you did in person. Dr. Oates is spearheading advances in using neuroimaging to personalize how we use brain stimulation techniques to treat mood and stress disorders. And you'll hear some of the great work today. He uses evidence from basic neuroscience to understand the mechanisms of brain stimulation and how and why it's effective as a treatment. And in parallel, he uses these insights and his clinical expertise to continually optimize the clinical applications of his work. On a personal note, I am a big admirer of his research and his broader contributions to the field. I first met him when he was here at Stanford during his early career, and I'm very aware that he's sincerely dedicated to making a difference in the lives of those impacted by psychiatric disorders through the best quality science. And he is a much sought after mentor and has contributed to the careers of many joining us today, as well as many in the field. So today, Dr. Oates will be speaking on a critical topic, making imaging relevant to precision approaches in psychiatry. And please join me in welcoming him. Welcome.
Thanks a lot. I'm really happy to see you guys. So some of you may know that I was a trainee there until about 8 years ago. And so I had some really great career development workshops at the VA with Ruth O'Hara, I know is still there. And I especially want to express gratitude for the connections and the advocacy that Lee has made for me in my career. They've been fundamental. So I'm really grateful to have a chance to come back virtually and talk with you guys. So yeah, if some of you saw the SOBP talk, there are a few new slides, but a bunch of the same information also that I hope will still be relevant and interesting. So one of the problems in our field, so coming up as a neuroimager in psychiatry, there are a lot of these kind of recent hits against our field and calling into question whether neuroimaging is actually useful in psychiatry. So replication problems, reliability problems, from the perspective of a clinician in psychiatry, they don't make any use of functional neuroimaging to do their jobs, to treat their patients, even though we've spent many decades and billions of dollars doing imaging studies. So where can we maybe start to build stronger, better connections? I think that's worth considering. And also that looking at bigger and bigger databases of patients may not always yield something really important or definitive. In fact, large studies don't always, even with their huge boost in statistical power, aren't always able to find some reliable biomarkers that we can hang on to and try to make use of in psychiatry. Also say that when we take these really beautiful imaging databases and we want to use them to in the treatment of an individual patient, that's where eventually we all want to go, right? And so the problem with using imaging published studies to use and make any kind of progress with an individual patient is that these are usually averages. They make really beautiful pictures. But if you try to take that and then apply it to an individual patient, there's not a straightforward path to doing that in a way that could facilitate their actual clinical outcomes. We've known about this for a long time in the imaging world. Like there's this Deanna Barch paper, more than 10 years old now, says, what if you compare these maps? So you look at the average map for this very like rudimentary, basic, well-trodden task, this working memory and back task. Say, okay, this is what the average one looks like on the set of images on the left. And this is if instead you use the lens of how many actual individual participants had strong activation in these regions, you can see that it shifts a bit, right? And I would argue that the map on the right is more relevant to the individual patient that you might see and you might be designing some treatment protocol around. But we usually ignore those images in favor of the ones on the left. Look at this new condition we found and where it's represented in the brain. Another problem that a lot of the very quantitative people, I think, need to be reminded of is that our ability to agree on a diagnosis for an individual patient is somewhat sketchy, and that gets in the way if you take really nice databases of patients and you say these patients have major depressive disorder, these ones have PTSD, and you assume that those labels are bulletproof and you use all of your metrics to try to reify the diagnostic labels, which are in themselves not really as good as they could be. You do better if you capture individual symptoms, but with these labels that they can just fall short of, or they could be different ways of asking the questions that clinicians may not agree so readily. So that's something that we can't ignore in psychiatry. Also say that you have a distressed patient, right? You apply a cutting edge treatment. They get better. A lot of them get better. What if a lot of them get better, like the Stanford protocol? That's amazing. That's super important. We really love that. But not everybody gets better, right? Some patients may even get worse, and some patients don't get much benefit. And if all you have is the clinical evidence, it's really hard to make progress beyond the group of patients that are doing really well. So if you don't capture some of the individual variations, in this case, the brain stimulation treatment, I would argue a brain measurement would be the most relevant aspect to figure out what can we do for the patients who don't respond even to this really fantastic treatment. So one of the ways that we try to capture some of these individual variations and understand what happens in the brains of people who get brain stimulation is to apply TMS in the MRI environment, which is something that I started doing in Amit Etkin's lab when he was at Stanford and I was at Stanford, and we set this stuff up together, and it was really challenging, but a lot of fun. And I'm still doing that today. And so that's what our current setup at Penn looks like. You can actually deliver TMS in the scanner, and you can engage circuits and see what happens in that person, in that individual person, in response to a TMS pulse. So the way this works for those who are imaging and fMRI aficionados is that you can send a pulse of TMS into somebody's brain, and you can capture the brain responds to that pulse, even though sending the pulse at the same time that you're making a recording would corrupt that image. And the reason is that bold is kind of slow, right? If you flash a checkerboard, if you have an event, capturing the brain response to that takes seconds. And so we capitalize on that with interleaving the TMS pulses and capturing what happens downstream throughout the whole brain in response to a TMS pulse. So just like any other event-related design, we do a whole bunch of those samples to an individual circuit, we average them, and we consider that the brain response for that patient through that circuit. So started doing this, as I mentioned, in Amit's lab. This was our first attempt at trying to hit individual nodes, so single points in a network for canonical resting state networks, pinging those circuits, sending the pulses through there and trying to engage the circuits. And we found some evidence that you can stimulate, you can engage networks, distributed networks, even though TMS is typically applied only to one spot, to one node in the network. So by hitting the right ones, hitting an accessible node of that network on the surface of the brain, you can engage downstream distributed networks with some degree of specificity. So building on that, since coming to Penn, thought, all right, there's some really juicy, interesting targets for depression and anxiety that we want to see if we can engage with TMS. You can't do it directly because these are like deeper in the brain, but through this networked approach, where parts of the network are on the surface and accessible to TMS, can we actually engage those deep brain areas? And let's show evidence that we can, again, at the individual subject level. So this is our basic design. We have fMRI readouts periodically with single pulses to ping the circuit to show what gets engaged downstream in response to those TMS pulses. And then we generate these average maps that show you all the downstream partners that come online in response to TMS to that one cortical spot. And when we do TMS outside of the scanner, we can still use the imaging data that we collect from that patient and then line it up with a camera, mark some fiducial points on their skin, and we can know where on that person's brain we're stimulating. So as long as you have some imaging data from that patient, it gives you a bunch of options to try to stimulate different brain networks. So if you have your own theory about how is the brain kind of divided into a parcellation, a network, what's more meaningful, task arresting fMRI, or a DTI, et cetera, you have some hypothesis about where is an important node in a network to engage it. You collect some imaging data and you can go to work doing experiments, behavioral experiments, treatments, targeting that brain area. All right, so we did that with the subgenual anterior cingulate cortex, which has been a focus of deep brain stimulation and TMS studies for a while. We started out doing this in healthy subjects and said, all right, if we use resting fMRI to we stimulate on the cortical surface, can we show some evidence that we can evoke a response in the downstream subgenual cingulate, which is what we were aiming for? And we showed we can, and that there's some degree of specificity for that. If you're focusing on a high connectivity pathway with the subgenual, you get more engagement of the subgenual cingulate compared to a control site like the motor cortex. And so we did that. We replicated in another sample, and in this time we're trying to see if we hit some of the positively correlated spots on the cortical surface and some of the anti-correlated or negatively correlated spots. Does it make any difference in terms of the downstream response in the subgenual cingulate? So we did our interleave design with those single pulses and averaged them, and we looked in healthy subjects, and we found that both of them actually do a pretty good job, whether you're hitting a positive connectivity or anti-correlated spots. Both of them do pretty well at engaging the subgenual cingulate cortex in healthies. But we brought in some depressed patients and said, okay, if the anti-correlated spots do still engage the subgenual, you can see that blue marks over there to the left, but the red ones, the positive connectivity, does it even better. So if what we really want is to reach the subgenual cingulate from prefrontal cortex And that's the mechanistically important target that we should engage in order to treat depression, which a lot of us think maybe is the case. Then following the anti-correlated targets may not be the best you can do for patients. So we've been following that up in a variety of different ways that I'll tell you about in a bit. But that's not the only target, right? We're also thinking about the amygdala. We showed that we can engage the circuit with amygdala connectivity pathways in a small cohort of healthy people. And we replicated that in a larger sample for which we also got some tractography data from DTI. And we wanted to see, is this like, does it seem like there's a direct pathway between ventrolateral prefrontal cortex and the amygdala? What evidence is there for that? And we found that we could engage the amygdala, show a nice evoked response. And we also showed that the fiber density for the putative pathway between where we stimulated on the cortical surface and the downstream amygdala, the fiber density of that pathway was associated with the strength of the evoked response in the amygdala. So we think that could be part of the mechanism of how TMS makes its way down to the amygdala. Say, okay, these are kind of nice little tricks. You can show some circuit engagement, but maybe some of these things are relevant to clinical outcomes and some of them aren't. Like, what's the evidence that these pings of these circuits is even something important when it comes to doing repetitive TMS, which is what we usually think of with TMS as treatments where people are getting thousands of stimulations and trying to improve their symptoms in psychiatry. Like, what's the relevance? Well, one of the things to consider is that it takes a really long time. It takes really a long time, even if you're only doing 5 days worth, but you're doing 10 sessions per day. That's a big burden on the patient and the provider. It takes too long, right? It takes too long to figure out, does this circuit work for whom? How long does it last, et cetera. We want something faster that we think is maybe just enough to push the symptoms in the right direction so we can link up all our measurements, the TMS fMRI and the outcome and clinical change. Look what happens afterwards, the TMS fMRI signal. So we settled on this 3-day protocol and we used the imaging data as our priority. We want to show that we can engage that circuit. We want to show that we can modulate the circuit. And instead of this correlational stuff where we just say, oh, look, the resting connectivity changed or it didn't, or a response to a task, we want to know, like, through the circuit we aim at, we want to show this causal argument. You ping the circuit and evoke something downstream in the brain. So not just blobs, colored blobs all over the brain. Short protocol, one short protocol. We didn't have a sham in this case, but we showed some evidence of circuit specificity because we engaged a couple of different circuits, and the patients themselves, when they're in scanner, you kind of move the target. They don't know which one is their intervention target or not, so you can show some degree of specificity with the target you're aiming for. So we did this design where we collect the baseline imaging data. We use that to make a target positive functional connectivity to subgenual. We put them in the scanner again and try to engage that circuit with TMS fMRI. Then we do a 3-day out of scanner intervention with intermittent theta burst through that subgenual pathway. And then we put them in the scanner again and ping the circuit. And I think we could learn a lot by this kind of design. The intervention itself is neuronavigated, so they have the camera in front of them showing where we're stimulating so that we maintain our target. And we did this low dose protocol for just the 3 days where we thought, we think this is enough to move symptoms and the circuit, and we just want to link these measurements together. And so we found that at pre-intervention, the strength of the ping, the strength of the brain evoked response in the subgenual predicted how well the patients would respond clinically. So we think it's important. This pathway is important. This is one way to demonstrate it. We engage the pathway more strongly in patient A versus B. Patient A is supposed to, if this is the right circuit, clinically respond better. So we show this relationship. And we think of this as their circuit integrity measure. We ping the circuit, The stronger that evoked response, the stronger the circuit between where we stimulate with TMS and the downstream subgenual. So we think this is meaningful clinically. We also showed that there's a relationship between the change in the evoked response in the subgenual, so TMS fMRI pre, TMS fMRI post, and the clinical response. So the bigger the change you have in that circuit communication, according to TMS fMRI, the stronger your clinical response. This is the circuit specificity. So we're like, okay, well, you stimulate the spot that has high connectivity with subgenual, but other spots in the brain are also engaged. So if you just looked at like the amygdala in response to pinging that place, is that relevant? Is that related to clinical outcome? It was not. Another thing you can do is you ping the amygdala circuit where we didn't apply the intervention, and you can see what does the subgenual evoked response to that circuit look like, and does that tell you anything about clinical outcome? And it did not, nor did the amygdala pathway, which the dark red one, which makes sense because that's not where we applied the intervention. But just to show some specificity, it's the subgenual evoked response and only the one in response to the ping where we delivered the intervention. So that dark blue line, that's the only one related to depression improvement. Anxiety also improved, right? So anxiety improved. This is the DAS-21. So this very like mood related subscale, and then this anxiety one is a lot about like anxious arousal types of anxiety. So both of these improved. The patients got better, but based on our subgenual evoked response, it was not correlated with anxiety improvement. It was only correlated with depression improvement. So like, okay, so the subgenual is related to the depression part. How did they improve their anxiety if it wasn't through the subgenual? And so I'll show you a slide on that in a bit. Also, we open the doors to everybody. We take all the patients who are looking for TMS to treat depression. And so after post hoc, we pulled out the treatment-resistant patients, and of course, they had more severe symptoms, but proportion wise, they responded just as well as the non-treatment resistant patients. So it's not a factor of how strong their depression is. Patients in both groups seem to benefit significantly from this intervention. All right, so back to the question of, well, subgenual couldn't be the only part of the brain relevant to their outcome, even though that's the one you were aiming for. And also, if anxiety improved and it wasn't related to the subgenual, like what stuff was actually changing in response to the TMS that was contributing to anxiety improvement. So we did this support vector regression of depression change onto the brain change to the evoked response, and same thing with anxiety. And as you can see, the subgenual comes up as a hotspot. If you look at the whole brain, like look everywhere, it comes up as a hotspot, which is good. It further validates our target. But there are a number of other brain areas, like in the hippocampus and over in the ventromedial prefrontal cortex, and some amygdala in the anxiety improvement map. And so since it's a new form of evidence of TMS fMRI, this may give you even better clues as to what new targets you might want to try for treating specific symptoms in patients. All right. Another thing we did was a clinical trial without TMS fMRI, just trying to build up clinical evidence of a more definitive dose of TMS over a longer time course. So we took 45 patients. They were all PTSD. There was a handful of MDD, and all the depressed patients had experienced traumas that precipitated or worsened their depression. So it's like a trauma-relevant depression and trauma-relevant PTSD cohort. And we used that subgenual cingulate pathway, our positive connectivity target, and we wanted to compare that to a scout-based target, so a lot less personalized. And this is one that people have used in the clinic before to treat depression. Fine motor cortex, go 6 centimeters ahead of that and say, okay, that's about DLPFC-ish territory for most people, depending on their head size. So we did this comparison. So we did intermittent theta burst to both of those targets. We have this kind of 2-session design per day where they're having an intervening task where it's supposed to kind of be a high cognitive engagement or a low engagement task, and in between 2 rounds of intermittent theta burst. So you do this Monday through Friday for 10 days. So 2 weeks of this protocol, and our primary question was, does that even matter? Does it, like, do people need fMRI? Does that work better than a scalp target? Amazingly enough, we don't really have a literature supporting that yet, even though, right, we know the same protocol is fMRI guided and it blows everything out of the water. It's so great and fantastic, and the clinical outcomes are amazing. We don't have this head-to-head comparison where we see that fMRI beats a scalp target or a structural-based target. So that's not in the literature yet, and it's kind of amazing that it's not. So we wanted to address that question primarily. And these are the clinical scores across the treatment and post-treatment. So we have the PTSD checklist, top left. The purple is the fMRI guided, the gray is the scalp target. The more negative you are on the y-axis, the more improvement you had in your symptoms. So as you can see for the PTSD symptoms, they're pretty neck and neck. They're pretty even, no significant difference. But on the PHQ depression scale, there was a more improvement for the fMRI guided compared to the scalp target. Now, it wasn't huge. It was a 60% improvement rather than a 50% improvement. But if you can imagine that degree of difference for doing the personalized targets, like making a difference in the patient's lives and their functioning and their outcomes, and may end up being really important, that it was a 60% improvement. And that's still a pretty nice improvement for TMS outcome. Also, if you look at some of the subscales of the PTSD checklist, there was one where the fMRI guided also beat the scalp target. And again, it was a little bit of an improvement, but statistically significant. So benefit for the fMRI guided and hyperarousal symptoms in PTSD. This is another way of looking at the data. So if you're to the left of that vertical gray bar, that means the fMRI guided is treating the patients better. So as you can see, like the PCL, it just has too much variability, but still the mean difference is better for fMRI guided. And you can see that the PCL, hyperarousal, and the PHQ are doing better for the fMRI guided. I also want to point out that this doesn't mean that fMRI guided is better than a scalp target, and all those ideas that people have out there from imaging studies, they have fMRI guided differences in patients, and we should use all of those to treat patients because they're going to be superior to a scalp-based target. Here's a nice big study that just got published in Nature Medicine showing that this fMRI guided target, which was based on functional connectivity of the anterior insula with the DLPFC, did not beat a scalp-based target that is used a lot in depression, the F3 EEG site. And that's a pretty good sample size for a TMS study, like more than 100 patients in each group. And they didn't even come close to showing a difference between the fMRI guided and the scalp target. Also want to point out that there are dose considerations. All the studies like throwing at you, they don't always have the same dose, and that could be a consideration. So for this study where they found no difference, they did 60,000 stimulations. Your guys' same protocol does 90,000. The data that I showed you for the PTSD patients was 24,000, so much smaller dose, and still we had a much stronger clinical effect than this 60,000 dose paper. Something that I think of for the future might be really fun to try is, what if you have multiple potential entry points for a network or for a downstream brain region at the individual patient level, right? We could ping multiple potential points and say which one of those engages the network or the region better for that patient. And the assumption is, based on our data, that would give you a stronger clinical response. So if you're like, what about for this person, the anti-correlated spot is better, right? Then I'm all for going with the evidence from the patient's own data to inform targeting. So I think that might be a really nice approach, but it's also hard to implement. Stanford, you guys can do this. We can do it at Penn. But if it's a strong enough signal that we kind of develop this evidence, maybe it can spread. So in conclusion, fMRI-guided TMS seems to engage the targets that we're after, the subgenual, cingulate, and amygdala. Also, the engagement is related to clinical effects. There's associations with clinical outcomes. If you take it before the intervention, if you show the change with the intervention, both of those are related to clinical effects we think are clinically meaningful. Also, the fMRI-guided TMS is more clinically effective than a scalp-based target. I'm not saying that everybody should use my target to treat depression. That's the best one, the only one, right? Probably you guys have even better targets that are going to work even better than the ones that I've tried so far. Absolutely, let's try it out. It's very unlikely that we've found as a field the best target for treating depression and that'll fit for every depressed patient. Likely we haven't done that yet, but we have something to start with, and I'm excited to push this target further. And I'll also make a case for adding imaging data. We learn a lot by having imaging data. So if you're going to do a big clinical trial and spend all that time and energy doing brain stimulation, I think we really need the imaging outcome data also to help us figure out better how it worked, for which patients it doesn't work, and to create new targets that will be even better going forward. Also say that there are other tools we're working with to say that how can we push the envelope to understand how brain stimulation affects circuits, how they get modulated, how to improve the effectiveness of the brain stimulation tools we have already. So we're working on these in working memory and worry and rumination specific targets. I think that's one of the things that the field has been talking about with some of the failed biomarker studies, is saying, well, maybe we just don't have a task fMRI data that are relevant enough to the patient's pathology. So maybe you need a smaller sample than thousands if you can actually do something that's more relevant to patients. And we agree with that, and we think that we might be able to get some leverage by having patients do individualized script-driven worry and rumination in the scanner. So I'll let you guys know how that works. Also, maybe resting fMRI is not the best targeting system for engaging a network. I'm open to trying other targets. We're also trying just some different ways of parcellating the individual data that have some theoretical background, like network control theory, that proposes that there may be more critical nodes or hubs in a network that should move the network more than other ones. So it gives us some like hypothesis for selecting among multiple potential targets. Deep learning approaches to especially blend some of the existing data that we have from the imaging literature with the individual patient level data to develop more reliable targets, but that are especially relevant to the condition we care about. And then collaborating with Casey Halpern on something that's very similar to what we do with TMS fMRI, suggesting that if you collect brain recordings from the individual patient, and you even try modulating them online and doing symptom provocation and things that are highly relevant to that disorder and to that person, that you might learn the most, the fastest. So I'm excited about pursuing that. Piloting new stuff. So one of the things we're going to start working on pretty soon is a controllable TMS where you can control the pulse width and shape. If you guys are interested in collaborating on that, and you have a machine or somebody there has a machine, we'd love to partner with you. Another thing that I think is that all of the clinics that do TMS should be doing research, right? Especially at academic medical centers. One thing that's very straightforward is to put in a task for them to do while they get TMS that you think is relevant to the circuit that could potentially boost their outcomes, that's like free to do. We can implement that. So we're working on doing that at a couple of different clinics. If you're interested, we'd love to do the same. And the funding for this kind of work is really great. If you've read some of the funding announcements from NIMH lately, for example, the R61, R33 grants, where they talk about evidence of circuit engagement as the precondition for the R61 to move on to the R33, that is such a perfect fit for TMS fMRI. All right, we can totally do that, and we have a pending grant that makes exactly that claim. Like we're like multiple targets, show circuit engagement, and then do the clinical, more definitive outcome in the second phase. So I think there's a lot of like motivation and excitement for this kind of work. If you're interested in learning more, I'd love to talk. And these are members of my center and my closest collaborators. With that, I will be happy to answer any questions.
Thank you so much for that talk. I have plenty that I could ask, but I'll go to the Q&A so I can let other people's voice kind of come out. There's a question, and I think this came up in kind of the slides where you're showing the dissociation between anxiety and depression in terms of some of the sites. And Gail Price was asking, in clinical trials, did you include patients with bipolar 1 or 2? And were there results that were yielded from that?
I haven't, but there's evidence suggesting that bipolar patients also respond well. And there's always a concern about, like clinically, one of the things to watch out for, the pitfalls of doing TMS, is to keep an eye out to see whether you induce some manic symptoms in the patients. And it's not like you cause mania where it didn't exist before, or that it has some enduring impact. But the clinical strategy generally is to just back off the stimulation, give it a bit of a break before you restart it. And anecdotally, that seems to work pretty well. But also, eventually, at Penn is treating bipolar patients with an accelerated TMS protocol, so that's in the works.
There's another question from Greg Salem. Amazing work as usual, Desmond. I really like the idea of using a task during treatment. Which do you use for depression, and has it yielded better results?
Yeah. So let me pull back up this slide. I kind of just forgot to mention this when the slide was on, but okay, this guy, so the bottom two rows showed a significant interaction between the task and the target for us. So if you were doing the 6 centimeter target, less individualized, you have a better clinical effect if you had the end-back task going between the 2 rounds of TMS daily. And if you did the fMRI guided target, you should have the relaxation condition. Those folks had better clinical outcomes, specifically on this avoidance subscale. And I think that something really basic like this is pretty straightforward and easy for us to work with. And Eddie Cheng and Catherine Scangos had a paper where they're kind of marching around and electrically stimulating a single patient and capturing what state she was in when they delivered the stimulation. And these data are kind of consistent with that. If there's this basic arousal dimension in conjunction with stimulating some of the targets, they may interact in a way that promotes mood change. And so these data are consistent with that. They have done, there are tests that you should stay away from and that people have used in the literature with hurting the clinical outcome. One of them was from some guys in Israel who did some depressive rumination during TMS, and that seemed to make the patients worse. So if there's a literature support for or against what you're doing, it's still really early days. And I say that all the tasks, even the OCD symptom provocation, which was part of the FDA approved protocol, we don't really know what happens when you don't do the symptom provocation, or if you have them relax instead of doing the symptom provocation. That's so easy to implement in a clinic. And so if you have a theory on what would engage the circuit you're aiming with at TMS, you should totally go for it. And if you want a collaborator or ideas, I'd love to talk about it more.
Thank you for that. That does seem like a lot of wide open territory. We have a question from Laura Hack. Thanks so much for the fascinating talk. Do you have any data on the durability of the effect of TMS in your trials and its relationship to target engagement?
Durability, I'm still piecing that together because we had a lot of patients fail to respond to our follow-up. So would we assume that they're doing worse and they don't respond at the 6-month mark, or do you think they're like doing better and they're too busy to respond to your queries? But I will say that there's decent durability. We'll probably have more durability if we have a bigger dose. So with our next protocol, we're going to go from the 2-week one to the 4-week protocol.
Awesome. I think Leanne has a question.
I jump in. Thanks so much, Desmond. This was fantastic, as always. I wanted to follow up. It's kind of a broader question, but relates to the ones that have been asked about, how you think of this way of engaging the circuits in a very personalized way and with different functions. I think that's so important. And the fact that you think about the pragmatics, like how would you implement this in practice? So my question broadly is, if you had a kind of like your wish list, like what would you like to happen if you think forward several years and we were using this more readily in practice? Do you have like a kind of vision for how you would like to see this implemented, and whether you'd have like for want of a better term, like a menu of these different kinds of stimulus provocations, or how you would think about
I think making the either the symptom provocation or the tools people use to online ramp down their symptoms, having both of these as priorities that we test out would be totally reasonable things to do. So you can take a basic CBT technique of like cognitive restructuring, ask the patients to do that, versus a session where you allow them to ruminate on their depression content. I think those kinds of studies would be completely reasonable. There's probably an interaction between what kind of stimulation protocol you run and the task that you have them do, but come up with a theory, test it. I think pointing out what doesn't help the patients can only go so far. We should have multiple active conditions in the trials to compare against one another. Something like the brain target by task interaction, I think that'll get us the furthest, fastest.
Thank you so much. I really like the idea of the multiple active conditions.
The sham, I don't know how much sham stimulation you guys do, but it's really like definitive in the scientific sense, but and the bar is higher to compare multiple active targets, right? But from the perspective of what can we promise the patient if we have multiple targets that we think are going to be at least somewhat clinically effective, it's so much of a like better position to be in as an investigator to say that some people are like having the stimulator turned off or just getting scalp stimulation. They're like, oh, that's pretty hard to sell to the patients who are really ill and they're desperate for treatment. It's very likely there's another active stimulation site that is really interesting that we could focus on. But again, like the difference between those is probably smaller. And so we need more patients to prove the difference.
Thank you.
There are not more active questions at this moment. I'm maybe going to take this opportunity to insert one of my own since we have a minute or two here. But just broadly speaking, I'm curious for your perspective, I guess above and beyond what you've already alluded to, where you think this is going in the next 5 to 10 years, what you're excited for in terms of developments of the field, since clearly it's developing quite rapidly. So I'm curious for your perspective as someone who's kind of keeping up to date on all of those developments.
There's some really boring stuff, like how to get a better TMS fMRI signal. It's really hard to get something under the TMS coil for a variety of reasons. And I think that would be useful for linking to outcomes. So we're playing around with some surface coils and some different stimulation protocols. Another thing that complicates the world of stimulation a lot is the controllable TMS that I mentioned. So there are ways of making stimulation more powerful without risking seizures still that we're toying around with that we want to test like immediately. And I think carrying those into behavioral interventions makes a lot of sense. And I like the mini intervention as a tool to test circuit hypotheses. So it's a lot more tenable for us to run a bunch of small experiments like that at a dose that we think is enough to change the behavior and iterate through a lot of different circuit hypotheses for modifying behavior. So I'm excited about continuing that part of the work, in addition to once we feel a little bit more confident that we're like, yeah, this is a good symptom related circuit and we know how to change it, then do the bigger, more definitive clinical trials that we think will have the most influence long term for the patients. So putting the measurement first in some research, putting the circuit hypothesis testing in another one, and then maximizing clinical outcomes. I think like having these inform each other and going on simultaneously in parallel should be really powerful.
I will also take this opportunity to ask a question. It's related to that you talked about actually getting not that good signal with the TMS coil. And a related question is, reliability has been a real issue in the fMRI field, people trying to improve the reliability. And I'm just wondering like how your thoughts are in terms of how reliability actually impacts the target engagement or finding the optimal target for TMS treatment.
Absolutely, super, super important. From one perspective, you want something that maybe fluctuates with somebody's mood symptoms, for example. So if connectivity to the subgenual like shifts around a little bit where the hotspot is, depending on how bad their symptoms are, it'd be nice to capture that. And it's probably important and meaningful. And then there's just like measurement noise, error sort of issues where you want to get the most definitive signal that you can in a given session, for example. So you might do multi-echo scans and do as long of a resting fMRI acquisition as you can because you want the most reliable signal that you can. And I will say that our protocols haven't made those changes yet. So the evidence I provided for you is like based on short segments of resting state, and the targeting should be further optimized, but what we've done so far may be good enough for determining a clinical target. But we're looking forward to updating our tools, our processing steps as the evidence kind of supports that. And I think we can only do better as long as we keep an eye on those things. I'll also say that the resting connectivity, like from our baseline, right, was not a clinical predictor, but we took another one the day before we did the intervention, and that one was a predictor of the clinical outcome. So there is some slop in there, and I think capturing the connectivity data closest in time to when you actually do the intervention there may be a benefit, at least that we've seen. But we also showed that connectivity from the correlation like is a predictor of clinical outcome, but TMS fMRI is a stronger clinical predictor. You put them both in the same model, the TMS fMRI explains the resting connectivity drops out of the model, no longer significant, and TMS fMRI is still significant. So I think we have something that's sort of relevant to the resting connectivity, but like captures a more definitive measurement relevant to the outcome.
Adam, did you want to, did you have other questions? I'm looking at our Q&A. I think we will invite people to jump in with any final questions. And this has been a great discussion. Anything else you wanted to highlight, particularly that you haven't been asked, Desmond?
You guys are awesome. I would love to work with you if we could figure out a way. I think the work you're doing is super relevant, and we all care about the same things. And I'm just really grateful to get a chance to hang out with you and talk about the ongoing work. I hope we continue the exchange.
Absolutely. Likewise.
Thank you.
That's a plan. Great. Well, thank you again. I'll ask Adam to wrap up, but we really